Hi, I'm Enrica Orsini, Education Scientific Lead at the EHA, and we are here today to speak about iron metabolism with Laura Silvestri. Laura, do you want to introduce yourself?
Sure. Thank you very much for this opportunity. I'm a senior scientist at San Raffaele Scientific Institute in the Lab of Regulation of Iron Metabolism. And I've been working to the basic mechanism that regulates hepcidin, which is the main hormone that regulates iron homeostasis since 2005. And I'm interested in particular in the crosstalk between liver function in related to the regulation of iron homeostasis and erythropoiesis function.
Very well. So we start from the basis.
Yes.
So do you want to illustrate us very quickly what is the role of iron in the body and in erythropoiesis?
Sure. So iron is an essential element since it participates to many biological processes, to heme production, to mitochondrial function, DNA replication, oxygen transport, and many others. So for this reason, it is essential, but it's a very reactive molecule. That means that if you accumulate iron in the cell, in the body, in the organs, this iron can be toxic. It can trigger reactive oxygen species, and in some pathological conditions, this can lead to organ failure. So this is the reason why iron concentration in the body should be tightly controlled, also because we don't have any active mechanism of iron excretion.
And this comes to the second question, that is, I guess this is the situation that will be very strict mechanisms that regulate iron homeostasis in terms of uptake, storage, utilization, and export. Can you give an overview of this?
Sure. So iron is absorbed by duodenal enterocytes, and thanks to these cells, we absorb dietary iron at a rate of about 1 to 2 milligrams per day. And these enterocytes are crucial, and they expressed a iron exporter named ferroportin. So thanks to ferroportin, iron enters in the circulation. And as I said before, since iron is toxic, iron should be tightly bound to transferrin. That is a glycoprotein that safely transports and delivers iron to all the organs and tissues. So most of the circulating iron is utilized by erythropoiesis for hemoglobin production and RBCs production. And indeed, we need about 25 milligrams of iron per day to maintain the basal rate of RBCs production, meaning that iron should be recycled, because we absorb only 1 to 2 milligrams per day, and the recycling of iron occurs through reticuloendothelial macrophages that degrades senescent RBCs, degrades hemoglobin, and release iron back to the circulation. We do not have any active mechanism of iron excretion, so the concentration inside the body should be tightly controlled, and the organ that plays this crucial role is the liver. And the liver indeed produces a hormone named hepcidin that binds and blocks ferroportin. In this way, hepcidin reduces the amount of iron that enters into the bloodstream by decreasing dietary iron absorption and decreasing the release of iron from reticuloendothelial macrophages.
So if I understand well, there is a balance between the level of iron in the organism and the level of hepcidin?
Exactly. So when iron goes up, then hepcidin should be upregulated in order to block ferroportin and reduce the amount of iron that enters in the circulation. And the opposite occurs when body iron concentration is reduced, as in the case, for example, of iron deficiency or iron deficiency anemia. In this case, hepcidin should be suppressed in order to stabilize ferroportin and favors the entry of iron into the bloodstream.
So if the hepcidin ferroportin axis is the central regulator, I guess there must be some mechanism that modulate hepcidin levels.
Exactly. Thanks to the study that have been done in the last 20 years, more or less, the mechanism that regulate hepcidin in the liver, and in particular in hepatocytes, have been clarified. So hepcidin is regulated through a pathway that is named BMP-SMAD pathway. The regulation of this pathway is mediated by several proteins. I don't want to go too much into detail, But for example, when iron concentration in the blood or iron concentration in the liver increases, then hepcidin is upregulated through a mechanism that activates this signaling pathway. The opposite occurs in case of iron deficiency, when hepcidin should be suppressed. And again, also in this case, we have a very important protease named matriptase 2 or TMPRSS that downregulates the pathway in order to stabilize ferroportin on the cell surface.
And there is a role in all this also from proinflammatory cytokines like IL-6, I'm thinking about the possible links with inflammatory states.
Exactly. So hepcidin is considered an acute phase protein that is upregulated by, as you said, by proinflammatory cytokines such as IL-6, but also IL-1 beta, although IL-6 is the main cytokines. And it plays a crucial role in, as you said, in anemia of inflammation or anemia of chronic disease, because in this condition, hepcidin is regulated not by the BMP-SMAD signaling, but by the STAT3 signaling pathway that is activated by proinflammatory cytokines. And it's a sort of defense mechanism that the organism activate in order to restrict iron to pathogens. So the problem is that the same mechanism that is used, that is activated in conditions of infection, is also conserved in the case of inflammation or chronic inflammation. So they share the same mechanism, that is the production of proinflammatory cytokines. And this can be, of course, deleterious, because when you have a high level of hepcidin, you restrict iron. So you don't have enough iron to sustain erythropoiesis and RBC production, and then you develop anemia or anemia of inflammation in this case.
And this is a clear example, going to the pathology from the physiology, how this regulated pathway can have a role in the pathogenesis of a disease. There are others. There are other diseases where the deregulation of the hepcidin ferroportin axis can have a role.
Sure. For example, in hereditary hemochromatosis, we know that mutations in key players in the regulation of the BMP-SMAD signaling and hepcidin production impair the ability of the liver to produce enough hepcidin, meaning that you develop iron overload because of ferroportin stabilization on the cell surface. And this causes the so-called primary iron overload. But there are other conditions in which hepcidin is chronically inhibited because of expanded and ineffective erythropoiesis. And this is the case, for example, of the so-called iron-loading anemias, such as beta-thalassemia, sickle cell disease, and in some cases also the congenital sideroblastic anemia. In this case, the expanded erythropoiesis inhibits hepcidin by releasing a protein, an inhibitor that acts on the BMP-SMAD signaling. And I think this is crucial because this underlines the importance of the crosstalk between two organs, the liver and the erythropoietic organs and the erythropoietic tissues in the bone marrow or in the spleen in the case of ineffective erythropoiesis. So the crosstalk between these two organs is beneficial, of course, in stress condition and physiological condition, but can be also deleterious in the case, for example, of ineffective erythropoiesis.
And going a step forward through the possible therapeutic application of all these, there are this deeper understanding of the basic mechanisms involved in the regulation of iron and hepcidin can help us to identify new therapeutic targets for these disorders.
This is crucial. I think that the basic research that have been done on hepcidin and the mechanism of hepcidin regulation and the regulation of the hepcidin ferroportin axis really help to identify the new therapeutic targets for this kind of disease. I can make an example.
Yes, sure.
For example, there is a genetic disease named, a very rare genetic disease named iron refractory iron deficiency anemia. in which the patients develop a iron deficiency anemia that is refractory to oral iron administration because hepcidin levels are inappropriately high. So the patients, even if you gave oral iron administration to this patient, the patients are unable to absorb iron because ferroportin is constitutively blocked. The gene mutated in this disease is TMPRSS6 or matriptase 2. That is a crucial inhibitor of the BMP-SMAD signaling. So, thanks to the study of this genetic disease, several researchers and scientists identified a potential therapeutic approach to, for example, hereditary hemochromatosis or beta-thalassemia that targets specifically TMPRSS6. So the first line of research demonstrated that in preclinical studies, so in mouse models, if you inactivate TMPRSS6 in a context of hereditary hemochromatosis or beta-thalassemia, you strongly improve the phenotype. Now, there are antisense oligonucleotides against the TMPRSS6 that are in clinical trials, there are siRNAs that are in clinical trials. There are some companies that are developing antibodies against TMPRSS6 because the scientists recognize the importance of this protease in the regulation of hepcidin. So this is very important because it really posed the basis for the developing of new therapeutic approaches starting from basic science. That is crucial, I would say.
And from, let's say, someone that is not really so inside the topic, the first idea would be, can we be able to directly work on hepcidin? What I mean, is it possible, for example, to, I don't know, administrate hepcidin or inhibit it, or there are obstacles to
Yes. No, the problem hepcidin has a very short half-life and it's a very complex molecule to be synthesized. It's very small but with a lot of disulfide bonds. But there are hepcidin mimetics, there are mini hepcidin. Hepcidin mimetics, for example, or hepcidin agonists are now in clinical trials. And for example, for polycythemia vera patients, it has been demonstrated that a hepcidin agonist is able to reduce the phlebotomy requirement of these patients in order to correct erythrocytosis. But there are also ferroportin inhibitors. For example, vamifeport is one of the ferroportin inhibitors that have been recently developed and that have been demonstrated to be effective not only in beta-thalassemia but also in sickle cell disease in preclinical models. So yes, we can somehow target directly hepcidin or the target of hepcidin that is ferroportin.
That is open a wide range of opportunities.
Yes, exactly. Yeah.
Thank you very much. It was very...
Thank you for the opportunity. Thank you very much.